Hello buddies. In this video I'll talk about flower development model, that is popularly known as ABCDE model of flower development. Arabidopsis thaliana is a model species. Why I am talking about Arabidopsis thaliana? Because it's a queen of model plants which is used in almost all the genetical studies due to its attractive characters. The flower development model was first studied in Arabidopsis. That's why here I am discussing about Arabidopsis. The properties of Arabidopsis, why it attracted many of the researchers, are 6-week generation time, small plant size, easy to grow and cross, a relatively small genome. It consists of 5 chromosomes and 125 MB genome size. It can be easily mutagenized. Genetic analysis of flower development. To understand the flower development, we have to understand different parts of a plant or different plants of flower. The flower development is enhanced by the auxin, which is one of the plant growth hormone. Due to the auxin stimulation, the vegetative primordia will convert to the reproductive or flower primordia that lead to flower development. Here it's a hermaphrodite flower which consists of petal, sepal, stamens, stigma and all those things. We can divide it into 4 parts, that is sepal, petal, carpel and stamens. Here I have given it. Sepal encloses and protects the bud. Petal attracts pollinator. Stamen produces pollen and carpel produces egg. Flower development. At a certain stage of plant development, the apical meristem switches program from a leaf producing to a flower producing tissue. At this stage, apical meristem becomes an inflorescence meristem. In inflorescence meristems form a number of primitive primordia that later develops into sepals, petals, carpel and stamens. This is a vegetative shoot apical meristem, and the side ones are leaf primordia. When it has to be converted to floral induction, it will lead to inflorescence meristem, the central one, and the buds which were stimulating the leaf primordia. Here it will be converting to flower meristems. In 1991, Coyne and Meyerowitz proposed the ABC model to explain how flower whorls develop in Arabidopsis thaliana and Antirrhinum majus. Flowers of most eudicot species are composed of 4 floral organs, which are sepals, petals, stamens and carpels. Carpels are gynoecium, that is female part, and stamen is androecium, that's a male part. These 4 components are all arranged in individual whorls around the meristem. Most of the genes of ABCDE model are Matchbox genes. This is the parts of Arabidopsis flower. Matchbox. It's a conserved sequence motif found in genes which comprise the Matchbox gene family. Matchbox encodes the DNA binding match domain. The length of the matchbox are in the range of 168 to 180 base pairs. Origin of matchbox: MADS. M stands for MCM1 from the budding yeast, that is taken from Saccharomyces cerevisiae. Agamus. A stands for agamus from the tail crest, that is Arabidopsis thaliana. Then D stands for dehiscens from the snapdragon, that is Antirrhinum majus. And finally the S stands for SRF, that is serum response factor from human Homo sapiens. In plants, Matchbox genes are involved in controlling all major aspects of development, including male and female gametophyte development, embryo and seed development, as well as root, flower, fruit development, floral organ identity, and flowering time determination. Signals to flowers. In order for flower to develop, two important genetic changes must occur. That is, change from vegetative to floral state and commitment to form flowers. Signals that indicate its time to flower is maturity of the plant, temperature, photoperiodism, and hormonal response. This is the flower development structure where different genes are involved. To explain it more clearly in the future slides, I have made distinguish the different genes differently. To understand floral development, researchers have to isolate single gene mutants that resulted in homeotic transformation. Homeotic mutations cause cells to develop into normal organs but in an inappropriate position. Homeotic genes help to determine the identity of tissues during development. Plant homeotic genes are, as I already told, these are matchbox genes. Arabidopsis researchers have identified several mutants with altered flower development. These homeotic genes are divided into three classes, A, B, C initially, and further they were divided into D and E. History of ABCD model. ABC model was given by Coyne and Meyerowitz. The War of Worlds Genetic Interaction Controlling Flower Development, which was published in Nature during 1991. After that, ABCD model was proposed by Colombo, Franken, and Kodze and other co-workers. They gave as the Petunia matchbox gene FBP11 determines ovule identity. This was published in Plant Cell. Then ABCDE model. This was given by Thiessen as development of floral organs identity, stories from Matt's house, that was published in Plant Biology in 2001. And finally, the quartet model was given by Thiessen in Nature in 2001. It's all about ABCD model of flower development. This model developed on the basis of Arabidopsis thaliana and snapdragon mutants. Most of the genes of ABCD model are matchbox genes, as already told. Class A genes consist of Apetula 1 and Apetula 2, controls sepal development, and together with class B genes regulates the formation of petals. That is taken from Antirrhinium lipless 1 and 2. Class B genes, example Pistillata and Apetula 3, together with class C genes mediate stamen development. Plus Antirrhinium. In Antirrhinium it is deficiens and globosa. That means Pistillata and Apetula 3 in Arabidopsis, and in Antirrhinium deficiens and globosa. Class C genes, that is ergamus, determines the formation of carpels. In Antirrhinium majus it is plena genes. The class D genes are seed stick and shutterproof, specify the identity of ovule in Arabidopsis, whereas in Petunia it is FBP7 and FBP11. In class E, that is sepalata, expressed in the entire floral meristem and are necessary, in that is SEP1, SEP2, SEP3, etc. Here I'll explain how the genes in coordination work together. Sepals means, as I already told you, E means it will be responsible for the whole flower development. So it will be with every part of the flower. So in case of sepals, A, that is Apetula 1 and 2, here we can see AP1. This is responsible for sepal development. For petals to develop, A, that is AP1, AP2, then B, that is AP3 and pistillata, then the E gene. E gene means sep genes. This is a complex which is responsible for production of petals. The third one is stamens, that is male reproductive parts. For the formation of this, B plus C plus E is required, and for this B means AP3, then C means AG, and E means sep genes, and this complex will lead to the formation of stamens. And for the carpel, that is female reproductive organ or gynoecium production, C plus E genes are responsible. Here C means AG and E is as usual sep genes. Finally, the ovules are developed. Ovules means the inner things which are present in the gynoecium, which are responsible for production of the seeds. And for the seed development, C plus D plus E. The genes are SHP for C, D means STK and AG, and finally E means sep genes. Likewise, for the development of sepals and petals, A plus B is required. For the development of petals and stamens, B, A and C, all the three are required. Then for the development of carpel, what we require is C, then ovules means it is D, and for the entire flower development, E along with all the four other genes are involved. That is the explanation to ABCDE model of flower development. So to understand the functions of particular gene, either it may be A, B, C, D, E, mutations were introduced in single genes. So in the wild type, all the genes were functional, and here in AP1 and AP2 were mutated, through which A was mutated. A was mutated means it was completely governed by C. And when AP3 and PI, that means B was mutated, it was absent, and the flower part was somewhat different. Likewise in different flower organs, the mutations were induced and their functionality was identified. To explain it in a better way, single gene mutants. First, to mutate Apetula 2. Here, petals transferred into stamens and sepals into carpels. These are called class A mutants. AP2 is required for concludes that AP2 is required for the formation of petals as well as sepals. Petals and sepals means the outer organs. And when AP2 was absent, only the stamen and carpel were present. Second is AP3 and pistillata gene, that is class B mutants. Here petals transferred to sepals and stamens transferred to carpel. That means AP3 and PI are required to make petals and stamens. That means when class B mutants were introduced, only sepals and gynoecium were present, whereas petals and stamens were absent in this. Third is ergamus. This is class C mutant. When class C mutant were present, stamens transferred into petals and carpels transferred to petals. So when stamens transferred to sepal and carpel to petal, that concludes AG is required for the formation of stamens and carpels. That means in the mutants, stamens and carpels were absent. That means male and female reproductive parts were absent, whereas only sepals and petals were present here when class C mutants were induced. Here after that, this is again the same explanation. This also indicates the same. Validity of ABCDE model of flower development by double mutants. Using this model to make predictions of what the double mutant plants will look like. Example, AP2, AP3 lacks both A function and B function, can't make sepals, petals, stamens. So only carpels were present, that means only gynoecium part is present. Only C functions here will be expressed in every whorl due to the lack of A function. Therefore, all whorls should look like carpels. This looks like carpel when AP2 and AP3 both were mutated. When AP3 and AG, that is lacking B and C functions, that means that can't produce petal, stamen and carpel. When it can't produce petal, stamen and carpel means only sepals are present over here. A function should be expressed in every whorl, thus sepals are produced in every whorl. This is the sepal in every whorl, that is AP3 agamous mutant. In ABC model flower development, triple mutant was induced that made lack of every flower part and the entire flower was looking like a leaf, or it's a bunch of leaves. A number of candidates for ABC class genes have now been identified. Representative example is class A is AP1, AP2, B is AP3PI and class C is agamous. Members of A and C class of genes are mutually antagonistic and both A and C class genes upregulate B class genes. A and C genes can also upregulate their own expression. Extension of ABC model experiment, UFO and ASK1 mutants. Two promising examples of this kind are ASK1, that was taken from Arabidopsis SKP1 like 1, and UFO, that is unusual floral organs, which are mutants in two components of SCF, that is SKP1 culin F box protein ubiquitin ligase complex. As a consequence of cell proliferation, the two domains move further apart until they separate. Auxin perception and transport in developing flowers. Pumping of auxins is mediated by members of the pin and aux families, with the former being auxin efflux carriers and the later influx ones. Disruption of either auxin transfer, that is corresponding to pin mutant, or its perception, that is corresponding to ARF mutant, can cause flowers to form abnormal structures. Utility of flower development model. Development of male sterile line, that is mutating B genes in Antirrhinium majus. Male sterile plants can be of greater use in hybrid seed production. Development of double flowers in ornamentals. Mutating C genes, example in Petunia and Antirrhinium, we can get the attractive petal type flowers and the double size of the flower, natural flowers. Controlling foot and seed shattering by mutating D gene. Development of unique flower form by mutating E genes, example green rose. If shatterproof 1, that is SHP1, and shatterproof 2, SHP2 are mutated, the seed pods fail to shatter or burst. They can be inserted into rapeseed or other cole crops to prevent pods from shattering. As we know, it's one of the major problem in rapeseed and mustard during the harvesting stage. The ABCDE model has been successful in explaining how a small number of regulatory genes acting alone and in combination specify the identity of the floral organs. Homologs in rice. Likewise in Arabidopsis, Antirrhinium majus or any other crop, in rice homologous of Arabidopsis ABC genes are Apitula 1 homologous genes are OS MADS 14 and OS MADS 15. For to Apitula 3 it is OS MADS 16. Agamus C, that is OS MADS OS MADS 3 and OS MADS 58. These are the genes which are analogous to the genes present in Arabidopsis. Summary. To summarize the whole ABCDE model, A is responsible for sepal and petal development, B to petal and stamens, C is for stamen and carpel, D is for ovule specification, and E is for the whole floral organ. And the two other mutants, UFO and ASK1 mutants for cell proliferation, and PIN and OX families for efflux and influx carriers of axons. Thank you.